Hi, I'm Michael Breakspear. I'm a professor at the University of Newcastle on Awabakal and Worimi country in Australia. I'll be presenting here a talk on metastable brainwaves as part of the whole brain connectome-based models of brain dynamics, an educational symposium at OHBM 2023, organised by David Mummy, Joanna Cabral, and John Griffiths. As an introduction, I just quickly show this video of a colleague and friend of mine, recorded actually while watching a movie, but I like to show that it emphasizes that even at rest or engaging in a common activity, humans are intrinsically dynamic with fluctuating psychological, physiological states. How do we model this? What kind of brain models engender this sort of spontaneous fluctuating behavior? My talk is in 4 parts: modeling complex natural systems. It's very commonly done, and it forms part of our daily lives. Then I'll move on to principles of large-scale brain models. I'll give an example of metastable brainwaves, which is actually the topic of this talk, and I'll briefly touch on neural field models. Okay, so imagine we wake up one morning, maybe in Montreal, we look out the window of our hotel room and we wonder, oh, well, here's a nice sunny day. But if I go out later in the day, should I go to the beach with a surfboard, or maybe should I go out with an umbrella? So how do I forecast or predict upcoming weather given the current observations? We can turn on the Weather Channel and do this quite trivially in modern life, or just Google weather. But actually, underneath these weather forecasts exists very sophisticated multimodal weather models run on some of the world's most powerful supercomputers. Weather forecasting is, of course, crucial to everything from daily activities, sports, ships, planes, spaceships, and weather crops, agricultural crops. And that's why there's such a busy business of models. Here's one on windy.com, just flicking through the various layers of these models: winds, waves, clouds, rain, etc. So how do these beautiful models emerge? What is driving them? Well, over many decades, there has been built mathematical, physical-based models of the weather. Here we see fairly early models parcellating both the atmosphere and the land and ocean into small regions, each of which obeys fundamental equation for the movement of liquid and fluid called the Navier-Stokes equation. These are derived basically from Newton's three laws. And they predict basically the movement of the atmosphere or the oceans under conditions of temperature and pressure. These models can be numerically estimated, but we need to feed them with real observations. So the world is covered in these weather stations and weather balloons and other means of recording all of these different observables that go into the model. And here we see such a model, which you can view through your Google browser. But the important underlying thing here is not just the physical modeling of weather, but the fact that the weather observations are constantly being fed into these models. So after the model is executed forward, then the forecasts are updated. This process of forecasting and updating observations is called data assimilation. So generative models lie at the heart of all models of complex systems. What about modeling the brain? Should we embark on this pretty standard approach in the natural sciences here in human brain mapping? Well, of course, just to reflect, We don't, the models are for properties of, in this case, liquid and gas. We discretely measure those, so we need a mapping from the states in the model to the observations. Likewise, we need a mapping from the states of the brain to observables such as electroencephalography and functional neuroimaging. So here we have the nutshell wrap-up of modeling the weather, and here we have, I think, what we are starting to do in computational neuroscience, using models to forecast observables, taking measurements and updating and refining the models. Now, of course, modeling has a long and storied history in neuroscience. In fact, in the 1950s, two neurophysiologists won the Nobel Prize for the Hodgkin-Huxley model of the individual neuron. This is a series of differential equations, capacitance-based, describing the flow of ions through transmembrane ion channels, which are gated by ligands and also by the voltage of the transmembrane potential. It's a remarkable series of studies with direct and detailed neurophysiological recordings, biophysics of membrane potentials of capacitors, and predictions to yield action potentials. Now, these models and simplified versions are extremely successful at the level of individual neurons, both not just generating a spike, but all sorts of things like regular spiking, intrinsic bursting, fast spiking. And there's a beautiful book by Eugene Isikovich that walks through the Hodgkin-Huxley model and all the various simplified derivatives. Of course, individual neurons are not what we study in human brain mapping, and they're actually immersed in functional subsystems and large-scale brain networks. Individual spikes are likewise really too weak to generally influence complex behavior. And if their precise timing was crucial, then their coding would be so high-dimensional and subject to random noise that behavior would arguably be unstable. And it seems increasingly likely, therefore, that brain function is based on what populations of neurons are doing and how populations encode in their distributions different states of the world or different states of other brain regions. For example, the local mean could embody the value of some observed state of the world, and the variance might therefore encode the precision in that observation. And this population activity is also matched to the apertures of fMRI, EEG, and MEG. So how do we do this? Here's a local neural population. I've integrated these with a reduced model called the Wilson-Cohen model. No, it's actually the Morris-Lacar model, apologies. And I've got here 800 excitatory and 200 inhibitory neurons interacting. These give rise to these spike clusters with time on the x-axis and neurons running down the y-axis and a dot every time a neuron fires. But if we take a brief window, we see underlying all these will be a simple distribution of states in these 1,000 neurons. And these distributions change in time. Now, a mean field approximation is basically to average over the actual complex distribution and assume that it accords with a Gaussian using something called the diffusion approximation. And here, as I just mentioned a moment ago, we could imagine that the mean encodes the value that the population is representing, and the variance or inverse precision says something about the precision with which that value is represented. Now, using an equation called the Fokker-Planck, I won't walk through it here, we can then propagate this distribution forward in time. And if this forward propagated distribution matches the actual distribution of states, then this mean field approximation is working. This is an approach used pretty systematically in physics. That's how we studied magnetism, superconductors, LCDs, queuing and swarming of animals. But in neuroscience, it's entered more recently. We can also get rid of this whole distribution and just look at the local mean. This is a neural mass approximation. So how do we do this? Here's some complex neural histology. We could model the whole thing with very detailed models of ion channels. That quickly becomes intractable. So then we might just take different compartments of the neurons, the dendrites, the somas, the axons, and the different types of neurons in the layers of the cortex and model those. We might reduce that further to something that just spikes and integrates. There's models for this, and there's software platforms for this. Or we might lump it all together and do a neural mass model, such as the Janssen-Ritt Wilson-Cohen model. Broadly speaking, going from the left to the right is what we are doing with a mean field approximation. These occur at every scale. A simple model of a dendrite might seem quite complex or quite simple, depending on your perspective, but it's a mean field approximation of ion currents through the ion channels. So now we're going to give an example of a neural mass model, something that I've played around with for 20 years now, and it models two interacting conductance-based populations, pyramidal cells and local inhibitory cells. And the pyramidal cells are conductance-based, and they have potassium channel conductance embedded in this model. I won't show the equations, but here's an example of integrating this model numerically. And in this particular combination of parameter values, we see a type of quasi-periodic flow called bursting. And this corresponds to 3 Hertz bursting, which is a common state seen in, for example, hippocampal tissue. We change the underlying parameters. We go through something called a bifurcation. We go from bursting to chaotic dynamics. And these sort of aperiodic excursions are also seen in neurophysiological recordings. So to go from a single mass up to something like the brain, we take individual nodes and then we couple them together and we allow their mutual influence through the white matter long-range connections. So here we have 2 interacting neural masses, one with a chaotic attractor, and then we can add many others with their own chaotic attractors. We inform these interactions by human connectivity data. We've got, you'll have seen in an earlier talk in the session, a whole brain network model. We can simply see what happens. In this particular simulation, we see whole brain wave patterns, rotating waves, something called breathers, traveling waves. So despite of the 500 nodes, each with 3 dimensions, a very high dimensional system, we see relatively simple large-scale dynamics emerge. And even though there's many different models and many different assumptions, this emergence of relatively simple dynamics from high-dimensional models is something that is common to most models in the field. The waves have been observed across different neuroscience imaging modalities, across different species, in perceptual tasks, in motor tasks, in anesthesia. And here we have a forward biophysical model that accounts for why they emerge. They don't come from nowhere. At the base of human connectivity data is what I refer to as a synaptic kernel, and that is this exponential-like function that ties the strength of distant neighbours according to their distance in an approximately exponential function. This means when you excite a node, that excitation is then seen by the node's neighbours across the whole cortex. And this is what pushes these large-scale waves across. Here's an example of observed waves from direct electrophysiological recordings. And here's an example of waves in this simple model. Now, I showed some different patterns of waves, and one might say, how do these different patterns emerge? Do you need different initial conditions? Do you need to push them through different bifurcations? There's basically a small number of dynamics through which a system can jump from one type of dynamic to another. There's a multi-stable system in which you really need to put noise into a multi-attractor landscape to get the system to transverse from one to the other, one attractor to another. A metastable system in which there's a series of weakly stable saddles, and here, even in the absence of noise, the system cycles from one metastate to another. Or you can have something called criticality, where there's actually a very weak attractor here at the origin, but in the presence of noise, the attractor is never landed upon, but the system shows long-range fluctuations. We've reviewed these different types of attractor landscapes in some recent papers. So what about our whole-scale brainwaves? To unpack these, we take the waves, each as a state. We look at the relationship of neighboring states to get this vector field, and we use the standard tractography algorithm to link these vectors into streamlines. Then we can look at the underlying architecture of these waves and see how they propagate in time. Show an example here, waves on the left, vector field in the middle, and the streamlines on the right. And what you see beneath the waves are relatively stable, if complex, nodes and spirals. During a wave transition, as we've just seen, there's a lot of collision between the spikes and the saddles. And this, therefore, is a form of metastability. We can then look at these transitions and partition up the time series and compare these to empirical data, which we've done. Sorry, I'm going to jump onto something else. But if you want to see the outcome of this, I refer you to Robertson et al. 2019. Finally, neural field models. These are models of very large-scale neural dynamics in which there's no longer a consideration of individual nodes, but rather the propagation of waves of activity across the cortex, treated continuously. As a motivation of what sort of patterns you can see in this, this is a common system that's been modeled by physicists called Chladni plates, after a German physicist, and a simple continuous surface vibrated at different frequencies, displaying these complex patterns of activity or modes. As you change the frequency, you change the number of modes that you excite. And the sound here is actually settling on the nodes between the peaks and the troughs in these waves. Vibrations on a plate obey a wave equation, depending on the stiffness of the plate. But once again, basically Newton's laws of motion, framed in the setting of a stiff metal plate with rectangular boundaries. These are the underlying Fourier-based modes. And when you drive the system with a particular frequency, you excite a number of these modes. This approach is very similar to modeling of the atom by the Schrödinger wave equation, where you get a series of modes, spherical harmonics, that capture how the electron in an excited hydrogen atom can be expressed. Boldly and recently, neuroscientists, theoretical neuroscientists have suggested that this might be a convenient way of modeling large-scale brain activity. So just as we get propagation of a quantum state as a particle through space, we can likewise consider the propagation of a perturbation on the surface of the cortex across the cortical surface. And you can read about this in Pang and Aquino et al., just published last month. The equation is a little bit more complicated than the simple one I showed. It's a damped wave equation. And from the spatial component, you can derive these spatial eigenmodes. Now, in the time domain, previously, Peter Robinson and I and colleagues have used this model as a model of resting state EEG data and as a model of epileptic seizures. These are from the considering global modes and looking at the time dynamics. But in the last few minutes of this talk, we're going to be talking about decomposing modes into spatial patterns and how we can do that. But basically, we pull out of this equation something called the Helmholtz equation. From the Helmholtz equation, we can derive eigenmodes using the Laplace-Beltrami operator. These are the low-order modes, and then there's a series of higher-order modes of gradually increasing complexity. Modes can be basically, or brain states can be summarized as a combination of modes and their activity. So M being the modes, F of T being the activity they support, and we can add some observation noise. So as we go to higher and higher modes, we get higher and higher spatial frequencies, and these support faster neural oscillations. Now I'm going to show an application of this approach to a clinical setting of neonates born extremely preterm and full term. We had scalp EEG data on these babies. We did source reconstruction, reconstructed functional connectivity networks, and then express these networks as a combination of eigenmodes using neural field modeling. This is the main effect of sleep. So these babies were either in active sleep or quiet sleep. These are thought to be precursors of REM sleep versus deep sleep. You can see as babies fall asleep, there's into quiet sleep, massive increase in long-range connections, which are shown here in blue. We can look at then a main effective sleep contrast in the top row and a main effective group. This is contrasting preterm and full-term infants. And we see in the preterm infants greater connectivity in this large-scale network. So this is a network view on baby electrical activity. These are the sort of different contrasts we get when we look at the strength of the different networks. So that's a network perspective. These changes in network connections preempt visual and social outcome at 2 years. And when we were looking at this, we decided to see if we could express these networks as a summation of eigenmodes. We just in this paper, chose a few low-order modes, and we allowed them to carry the alpha frequency that we saw in the babies. So this is how the model works. We take cortical eigenmodes, we allow them to be carriers of high-frequency activity plus noise, and then we look and match the parameters of the model based on their projected functional connectivity. So on the left is the data, and it's a kind of general linear model, but the model is based on eigenmodes and their carrier frequencies. We can solve this for each baby, and then instead of getting changes in network connections, we get changes in eigenmode expression. And these show quite different patterns from the network, but they are basically projecting the carrier frequency into network space. There's significant group by sleep effect in the expression of the modes. So here's the modal perspective, and here's the network perspective on the same underlying data. As I preempted before, there's a more ambitious expression of cortical eigenmodes on adult human fMRI activation maps, and this approach works remarkably well. But I'm going to defer to talks that will be given tomorrow at 8 o'clock in an early morning symposium chaired by Alex Finito. And also, if you'd like to see me discuss neural mass models and neural field models, I'll be speaking in a symposium on Wednesday at 8 a.m. Resources for these simulations can be found in the Brain Dynamics Toolbox and the Virtual Brain. This research was supported by the NHNMRC and the ARC. And here's an image of my group at a recent retreat. So thank you for watching the talk, and I hope many of you are able to join me in person at OHBM.